The cell cycle is a series of events that occur in a cell in order for preparation of division. There are 4 stages in the cell cycle. The first stage is G1, in which cells increase in size. The second stage is the S stage, which is for DNA synthesis. The third stage, which is G2, is where cells prepare to divide. And then we have the M phase, which is the actual physical division of mitosis. G1, S, and G2 all combine to give interphase. Cells use special proteins and checkpoint signaling systems to ensure the cell cycle progresses appropriately. While in the G1 phase, the cell chooses whether or not to divide. Once it passes this checkpoint, it enters the S phase. There it'll look for damaged DNA, see if growth factors are working properly, such as molecular signals, check size being suitable to divide, and see if it has enough energy and nutrients to divide.
If the cell doesn't meet checkpoint requirements in G1, it may leave the cell cycle and enter the resting state at G0. If conditions improve, division will resume, and if not, it'll stay at the G0 permanently.
While in the G2 phase, the cell checks for any additional DNA damage, also for proper and complete DNA replication where it can produce healthy daughter cells. P53 is a gene that codes for a protein that regulates the cell cycle and therefore functions as a tumor suppressor. P53 works by binding to DNA and either activating or suppressing its expression. If damaged DNA is detected, CDK is inhibited and there's no cell cycle progression. This is kind of like a TSA checkpoint in an airport. If you don't pass security, you can't proceed. At this point, the cell is arrested for repair. If P53 is unable to repair the cell, it triggers apoptosis. If P53 is able to repair the cell, it will eventually proceed in the cell cycle.
If errors are detected in G2, the cell will pause here and allow time for repair. If damage is irreparable, the cell may undergo apoptosis. This is so the damaged DNA isn't passed on to daughter cells and is crucial in preventing cancer. If DNA is damaged along with P53 can't bind to it. Thus, the CDK kinase will activate and cell cycle progression will occur, and the damaged DNA will be passed on. For all of these reasons, this is why P53 is the guardian of the genome.
In the M checkpoint, also known as the spindle checkpoint, the cell checks if all sister chromatids are correctly attached to spindle microtubules.
In the M phase, since sister chromatid separation is irreversible in anaphase, the cycle won't continue until all chromosomes are firmly attached to at least 2 spindle fibers from opposite poles of the cell. The cell looks for floating chromosomes in the wrong place. If chromosome is misplaced, then the cell will pause mitosis, giving time for spindle to capture lagging or stray chromosomes.